Hello everyone, I want to welcome you to our showcase session entitled New Frontiers in Pharmacogenetics. And I want to go ahead and get started. I'm Kathleen Barnes. I'm relatively new to Oxford Nanopore, leading population health and precision medicine. And I'm really excited to be here with our collaborator from Twist Bioscience, Dr. Tina Han. I'm going to jump in and just do a very brief overview of pharmacogenetics. And then Tina and I are going to have a deeper discussion about what's new between Twist Bioscience and Oxford Nanopore. I want to just alert everybody this is not an online session, so you're welcome to address your questions after we have our conversation. We'll take questions from the floor, but we won't be able to use the event app. So jumping in, how do we define pharmacogenetics? Pharmacogenetics has been around for a long time. It's how a person's genetic makeup determines how someone responds to a particular medication. And pharmacogenetics, while it's been around for decades, it's really been spotlighted more recently for a number of reasons. So first of all, more than 99% of the population has a genetic variant that's recognized as a PGX variant that determines how you respond to a medication. Also, depending on that population, anywhere from 45% to 79% of a population will carry an actionable variant in the cytochrome P450 gene, or CYP2D6. And CYP2D6 is related to commonly prescribed drugs, analgesics, antidepressants, beta blockers. In fact, CYP2D6 accounts for some 20% of commonly used drugs. And to this end, we can argue that CYP2D6 is probably one of the most important pharmacogenetic genes. And more than 50% of emergency department visits are in fact attributed to adverse drug reactions, or ADRs, in the Medicare or aging population. And in the United States, ADRs are now the fourth leading cause of death. The impact and management of ADRs is over $30 billion globally. And finally, PGX is really relevant when you consider the fact that among healthy individuals are typically on medications in at least two PGX-informed genes. So the bottom of the slide that's highlighted here really summarizes the national bodies that help us determine what is clinically actionable, the most notable being the Clinical Pharmacogenomics Implementation Consortium, or CPIC, along with the FDA. So on the next slide, I'm highlighting that even though PGX has been around for a long time, pharmacogenomics, there are notable challenges in the implementation of pharmacogenomics. This isn't a comprehensive list, but I've tried to summarize some of the big ones. So first of all, concerns regarding clinical utility and the cost effectiveness. There's still debate in that field. Health system infrastructure and data management. It's one thing to be able to test a patient within a health system, figure out how you're going to pay for that, but it really is a significant lift for health systems to build out the clinical decision support, integrating that into the EHR, and ultimately building out the best practice alerts, or BPAs, so that providers are guided on how to act on that information. Of course, a major obstacle in PGX has been how to pay for it. And currently, reimbursement in most places is really limited to what we call reactive testing for one or a few genes for a very specific case, rather than preemptive PGX. And then finally, there are challenges with the platform itself, how we fully capture the PGX genes, again, coming back to CYP2D6. So on the next slide, I want to put an optimistic slant on pharmacogenomics and say, is it time for broader implementation of pharmacogenomics? And the illustration on the left is a summary from a relatively recent study that really demonstrates that we're beginning to hit our stride in implementing PGX. So for example, with clinical utility, we're better and better at determining that. the economic value of PGX, when you think about that $30 billion price tag of ADRs, user acceptance overall is increasing, and even implementation in health systems. And while this isn't widespread, there are certainly health systems that are really beginning to stand up PGX, particularly for specific use case populations. Importantly, and you're going to hear a little bit more of this from Tina, we really reached a point where the technology, the platform itself, is better and better for capturing PGX. Other factors that I've summarized on the right side of this slide is a robust global market. The fact that governments are beginning to insert themselves to insist on PGX at the point of care. So think about the NICE program here in the UK, moving towards implementing CYP2C19 testing for a new stroke patient, or think about the Right Drug Dose Now Act in the United States. A really compelling argument, if you will, for comprehensive PGX also has to do with our rapidly aging population when we think about polypharmacy. So polypharmacy being when a patient is on 5 or more drugs at a point in time, they are at a greater than 50% chance of an ADR if they are a polypharmacy patient. Incidentally, polypharmacy is the 5th leading cause of death for the older population. And in terms of reimbursement, we are beginning, slowly but surely, beginning to make some headway in reimbursement. When you consider, again, in the US, some 60 out of 134 CPIC recommended drug gene pairs can actually be reimbursed. And finally, there's just more and more compelling evidence that health systems want comprehensive PGX, again, as I mentioned, for certain niche populations such as the value-based population. So with that overview, I'm really delighted to introduce our guest to this showcase session, Dr. Tina Han. A little bit about Tina. She earned her PhD at the University of Maryland. Go Terrapins. She completed her postdoc work at the Max Planck Institute for Molecular Genetics. She led the Genomics Core Facility at the Institute of Molecular Biology in Mainz. And she was the team leader of R&D at CeGaT. Sorry. And in 2018, she joined Twist Bioscience as a field application scientist, and since 2020, she has been involved in the development of long-read applications. So, Tina, I'm going to hand it over to you at this point. We're super excited to be here together and share with all of you the press release of this morning that there's now a partnership between Twist Bioscience and Oxford Nanopore with a new PGX platform. And Tina, do you want to, we put this slide together to sort of underscore the high points of this collaboration and this new platform. Love for you to share with it.
Yeah, thank you again for the invitation. It was an honor to be here because most of the work has been done by the ONT team. So feel very privileged to present the data. So in this panel, there are about 49 genes. And then we cover all the 4 different CPIC categories, so from level 1 to level 4. So then you mostly find a lot of actionable genes, but we also include something maybe worth checking in the meantime before they become actionable. And then on top of that, you can also add the mitochondrial DNA, because there's also an actionable variant in the mitochondrial genome. So again, this is a very comprehensive way to interrogate all these druggable variants and looking at the high-throughput manner. And what Twist does is we make DNA. So our probes' DNA are double-stranded. So when we do any capture experiment, they are like, you have 2 shots in one go. Then you can capture more of the molecule that's worth sequence. And since our amplification method is very uniform, and it's kind of translating into the data, so there's no GC bias carry out in your capture data, or when you do CMV calling, it's just much easier just because the evenness of the coverage that brings it to the data. So when we work with Nanopore together, putting the the panel together with the long read, just a perfect match, right? Because right now, pharmacogenes have a lot of pseudogene in there. So when you have a read, you actually doesn't know where to map if you are using a short read sequencing. But now with long read, you can just confidently map, say, well, if it belongs to CYP2D6 or CYP2D7. So again, the nanopore team work on this, and especially with a tool called Chinook that you heard earlier from Cecil's talk, is that it can really highly reconstruct this whole gene alleles, and then you can also do CNV and diplotype analysis on top of it. And so maybe let's talk about how can you run on the nanopore instrument, all right? So right now, the lab has done is they test 8 samples in one ming ion flow cell and 48 samples in a Promethean flow cell. So really give you a flexibility to go from low throughput to high throughput. And you can also easily scale out, right? Because Promethean, you can also have a multiple flow cell running at the same time. And so now we are actually providing you one assay to replace maybe 5 array assay you have to do in the past to interrogate SMV, indels, structure variation, phasing, and on top of that, haplotyping. Now you can do all together. And since the beginning, I mentioned we also can have the option to put into the mitochondrial DNA. So in case you are interested in hydroplasmin, you can also just spike in a little more, so you get a 500x or 1000x coverage if you wish. So I think now we maybe talk about the software, so how to analyze data, because I think that's a huge bottleneck. So first of all, the nanopore team also announced yesterday, right? They had a software release of Dorado 0.7, so that can enable you to demultiply the twist data effectively. And also the team is working on the analysis, streamlined analysis pipeline you can use in the very near future. So probably lastly, we also have ongoing project to test on the Twist Alliance dark regions panel. So whatever you cannot get from short read whole genome, now you kind of can use this panel to make out the difference. And we also are in the process of testing Twist exome 2.0 because your sequencer is actually versatile. You can do both short and long read, so why not just also look at the short read whole genome?
Very exciting. That's awesome, Tina. Thank you. Just a couple questions from me to you, and then we'll open it up to the floor. One of the questions to ask you, the burning question is, what do you think the implications are for fully resolving CYP2D6, for example?
I think in the past, people are using amplicon type of approach. Yes, you can do long read amplicon, but then it's not scalable. You probably can just put 10 amplicon in one run, but then most of the time you may have to separate them. Otherwise, there could be a competing between the primer pairs. And so amplicon is not scalable. And array, yes, is cheap, but then maybe array is limited to the targets on the array. It's not easy for customization. But since our approach is we can make your panel in 2 weeks, right? So then you can just come to us and say which kind of genes you want to be included, and we just whip out the panel and ship it to you. Right. And you can test the next day. Yeah,
that's great. That's great. That's so helpful. Last question, and I promise to open it up to everybody. So how do people get their hands on this?
Yeah, so maybe before I jump to answer that question, I forgot to mention the input. How much DNA do you need, right? Normally, we saw long read sequencing, you need a microgram sample. But in this case, not, because we do capture, and we also have PCR involved. So you actually can start as little as 200 nanogram. And they don't have to be super or high molecular weight gDNA. We have tested on a lot of 20 kb of a sample. Because the first step in our protocol is shearing. So you can also just do a softer shearing or short shearing. Then you can still have a great result with 5 to 6 kb at the end.
Great.
And the whole workflow is less than 5 days. So if you are experienced with Twist protocol, you can finish in 1.5 days. And for sequencing, just 2 days. And then you can also analyze the data on the fly, right? So, so less than Big 5 advantage. Days, you get your data from sample to data.
So a shorter turnaround time. Yeah. Fantastic. Thank you. I'm happy to take questions from anyone on the floor. We have another 5 minutes or so. Anybody?
And lastly, we have a QR code on the slide.
Yes.
So if you can scan it, if you are interested in joining the beta program, yeah, just fill out the detail. We will get back to you.
And I was also to remind everyone to please see the poster in the poster session.
Yes, so there's a poster right there across from here. And we also have a virtual pool. So the Twist Bioscience, we also have a copy of poster in case you miss it. Yeah.
Thanks. So any questions from anyone? Sure. Do we have a mic?
The mic is coming.
There we go.
Hi, thank you. Could you develop a bit on Chinook and if it's open access, how it works, if it only works for those pharmacogenetic genes or if it can be open to any other genes? Thank you.
I think Chinook right now we have tests on CYP2D6.
Correct.
Primarily.
Correct.
And then I heard the tools will become available soon.
Right. So it's coming.
It's coming.
So we
But then
answer.
sign this up, right? Fill the form and then express your interest and we can get back to you.
Thank you. Any other questions? Okay, here we go.
Oh, there's also one.
Hi. I was wondering about your barcoding, because obviously initially with the Illumina side of it, it's quite short barcoding. Will you be editing your barcode panels for more nanopore, so more functional for the nanopore side of things with barcoding? And also, will you be switching up your adapters so you can have the nanopore adapters as part of the capture and reduce some of the lab time as well with that?
So we actually don't use the typical nanopore barcode. We are actually follow our Illumina workflow. This means we are using a truncated Y-shaped adapter to do the ligation first because this is more efficient. And then we add our UDI, so we have a 10 base pair right and 10 base on the left. So a total you have 20. So that's enough for the accuracy. Yeah. And then the Dorado is able to do that. So you can give it a go.
Any others? Well, Tina and, sorry, there's a question.
Right?
Yeah.
The 5 days turnaround time that you mentioned, is it for from DNA till data or?
Yeah, so for extracted DNA, and you go through the library prep, the twist part, and twist enrichment. Normally it's overnight, and next day you just do some capture wash, so it's only half day. So theoretically, you can also start doing the nanopore library prep and then start sequencing. So then you can also, create some shortcut once you become very familiar with the workflow. So even you can shorten to 4 days or less than 4 days. Yeah.
Good question. Any others? Well, Tina and I will be around to take any other questions. I really want to thank everybody. Thank you, Tina, for joining us at this event. And thank you again, everyone, for being here and letting us share our news with you. Thank you. Enjoy London Calling.